question 36. In this question we are given with 4 statements and we have to tell which statement is right. Now in this the first statement is in transcription adenosine pair with uracil. Now we know that in transcription it is the formation of RNA from DNA. So we have that in RNA instead of thymine there is an uracil. So whenever the transcription occurs, so in front of adenosine you will have uracil. Okay. So this statement is correct. Second statement, regulation of lac operon by repressor is referred as positive regulation. But we know that in case of repressor the regulation of lac operon is negatively regulated, it is not positive one, because the binding of repressor to the DNA will stop transcription, and that is why we call it as a negative regulation. So this statement is wrong. Then third statement, the human genome has approximately 50,000 genes, but as our finding it is clear that the human genome contains around 30 to 40,000 genes. So this statement is also wrong. And the fourth statement, hemophilia is a sex-linked recessive disease. The gene of hemophilia is present on X chromosome. So that is why it is a sex-linked recessive disease. So out of the 4 statements, the statement first, that is A and D, are correct. So our answer is third. Question number 46. In this question, diagram of polymerase chain reaction PCR cycle is given, and we know that the PCR cycle consists of 3 steps. The steps are A, first step denaturation, then second step annealing, and the third step is the extension. So PCR is a 3-step thermal cycle in which the first step is the denaturation. In this denaturation it is heated up to around 98 degree where both of the strands get separated. Then in second step we have annealing where the primers are attached, and in third step extension where the take polymerase, the heat resistant enzyme, proceed the synthesis. So now we have to given some matches here. Now option number first have C, the extension in the presence of heat stable DNA polymerase. The C step is extension, and in this step the enzyme DNA polymerase is used. This is the correct option. Then second option, this step A, annealing with the 2 states of primer, but we know that the A step is the denaturation. So this option is wrong. Third one, B, denaturation at temperature of about 98 degrees separating 2 DNA strands. This is not B step, it is the A step. So this one is also wrong. And the fourth one, A, denaturation at a temperature of about 50 degree. Of course A step is denaturation, but the temperature is not 50 degree, it is 98 degree around. So the fourth option is also incorrect. So in this the answer will be the first one, the first option C, extension steps. Now question number 47. This question is for बायोलिस्टिक. बायोलिस्टिक एक डायरेक्ट जीन ट्रांसफ़र की टेक्निक है जिसमें बिना किसी वेक्टर की हेल्प से हम सेल में डायरेक्ट जीन ट्रांसफ़र करते हैं. बायोलिस्टिक को हम जीन गन भी कहते हैं. बायोलिस्टिक also called as gene gun. Now in this method some particles of gold or tungsten coated with DNA are bombarded on the host cells, and that is why, because it involves the bombardment of particle, so the host cells must have the cell wall around them, some protective covering, so that it can bear this kind of bombardment, and that is why it is being suitable for those cells which is having some protective covering like cell wall, and that is why it is used for transformation of plant cells. So in this question we have the 4 options. First one is the constructing recombinant DNA. It is a direct gene transfer method, no rDNA will form. Second, DNA fingerprinting, in this it is not used. Then third, disarming pathogen, in this method also it is not used. It is the method which is used to transfer the gene in the plant cell. So that is why the fourth option, transformation of plant cell, will be correct. Question 48. The 48th question is, a test cross is carried out to. Test cross is the very fundamental cross of the genetics. This cross is used to find out the genotype of any organism having dominant character, and it can be performed at any level, means any generation, maybe at F1 generation, at F2, whatever. So now in this question it is carried out to among the 4 options. It is the third option where we have the determine the genotype of a plant at F2 level. This will be correct. The other options to assess the number of alleles, to determine whether the species will breed successfully, or to determine the linkage will not be appropriate for the test cross. Test cross is mainly done to find out the genotype. So the answer of this question will be third one. Question number 16. Now in this question, the question is being asked that the tobacco plant resistant to nematode has been developed by the introduction of DNA into the host plant. Now this particular technique, this entire question is based on a very novel approach called RNA interference. RNA interference technique, or RNAi. Now by this technique the nematode resistant plant have been developed, and in this DNA is being transferred into the tobacco plant. This DNA, when it is introduced into the host plant, it produces 2 types of RNA, the sense and antisense RNA. This RNA combine together and form double stranded RNA, and this will start interference, and it will prevent the growth of the target gene. So according to this, the question, in this question we have the third option which is appropriate, that this DNA will produce sense and antisense RNA. The first option an antioxidant, second option a toxic protein, and the fourth option a particular hormone. These things are not produced by the DNA which is being transferred in the host plant. It is the RNA, the sense and antisense RNA, which is being produced by the DNA. So the correct answer will be third. Question number 21. Now in this question, in genetic engineering the antibiotics are used. Actually in this rDNA technique or in genetic engineering, antibiotics are generally used to select the recombinant from non-recombinant. Actually we use a vector here, and for the vector we have the 3 important property. First one is the ORI, second one is the presence of selectable marker gene, and the third one is the presence of cloning site. So for any vector these 3 properties must be required. Now when we transfer a gene with the help of vector, after that we have to use this vector to select the recombinant and non-recombinant. Now in this, antibiotics are used in the steps when the selection is being made. Our question's first option, as a sequence from where the replication start, actually it is the ORI sequence, and that is in vector from where the replication start. In this case antibiotic is not required. Then to keep the culture free of infection, for that normally we require the sterilization procedure. For that no need to use antibiotics. Third one, as a selectable marker. Actually it is the selectable marker gene. In this case we generally use an antibiotic resistant gene, and it is used to select the recombinant and non-recombinant. So that is why this one is the correct answer. Fourth option, to select healthy vector. This is not the method. Antibiotics are not used to select the healthy vector. Vector selection methods are different. So the question number 21, the correct answer, the third option, as selectable marker.